Are you a fresh graduate? Do you want to fulfill your educational aspiration by pursuing higher education in various interdisciplinary and multidisciplinary areas like environmental science, environment and occupational health, philosophy, sustainability science, folklore and culture studies? If yes, the School of Interdisciplinary and Transdisciplinary Studies offers various programs like Master's Program, Graduate Program, Postgraduate Diploma, Certificate and Appreciation Program. The school offers 5 Master's Programs. Master of Science, Environmental Science. The eligibility criteria for MSc Environmental Science is Graduate in Science, BTech, BE, Agriculture Graduate, Forestry Graduate and Veterinary Graduate. School also offers Masters in Environment and Occupational Health, Masters in Sustainability Science, Masters in Philosophy and Masters in Folklore and Culture Studies. The eligibility criteria for these 4 Masters programs is graduate in any discipline. The school also offers various socially relevant and need-based PG Diploma programs like PG Diploma in Sustainability Science, PG Diploma in Environment and Occupational Health, PG Diploma in Folklore and Culture Studies and PG Diploma in Migration and Diaspora Studies. The school also offers PG Certificate in Climate Change and Avshist Prabandhan mein Praman Patra. The school also offers 2 doctoral programs, PhD in Environmental Science and PhD in Interdisciplinary and Transdisciplinary Studies. The school offers various innovative technologies and multimedia for providing a very excellent student support system. The study material is of high quality. The learner support is provided through Gyan Darshan, Gyan Vani, IRC, Swayam Prabha and Swayam channel of IGNOU. For more detail, please log in to IGNOU website. For online admission in these programs, visit school site and also the Samarth online education portal of IGNOU. So please join these programs and fulfill your educational aspiration.
Hello to all the viewers and learners of Gyan Darshan. Myself, Dr. Ravi Rajwanshi from Discipline of Life Sciences, School of Sciences, IGNOU. Today I am going to speak about a topic from your unit on recombinant DNA technology, where there is a section called Methods of Genetic Engineering. This is a course from BSc Journal, and the name of the course is Economic, Botany and Biotechnology, bearing a code BBYET143. So, today I am going to cover some of the important topics like transformation, including bacterial transformation, agrobacterium-mediated transformation, direct transfer of gene of interest, and a very latest technology which is always in the news also these days, that is CRISPR-Cas9. To understand the topics related to gene transfer, first of all, we have to understand what is exactly transformation. So, transformation can be defined as a process where exogenous genetic material is taken up or incorporated into a particular cell through a cell membrane. This process can be done in a laboratory also, whereas it can occur naturally also. We will see how this process is taken up in laboratory condition by learning from the nature itself. To understand the topic of transformation, we should also understand that what type of cells can actually take up this genetic material. For that, you need to have a cell which is competent enough to take up this genetic material. Sometimes we also call them as competent cells. So what is actually the competency? A competency can be defined as a state where time limited and a particular environment condition is given to a particular cell when it is capable of taking a genetic material from outside. So, this process basically you can see as a very important process for the technology of recombinant DNA technology. And you can say even this process is a backbone for generation of GMOs, that is genetically modified organisms. So let's start with this topic in detail. To understand this topic, first of all, we will take up a simpler system that is called bacterial transformation, because it's a unicellular organism. So anything which we have to start with is from the less complex form. So in case of bacterial transformation, the first step begins with preparation of competent cells that needs to be transformed, and this usually require a bacterial strain which is usually stored in a liquid medium and as a starter culture, and these strains are basically made competent through a process. There are different processes like heat shock or electroporation. In fact, you can also use some chemical treatments. There is a series of treatment to be given which includes calcium chloride, So these treatments can make a cell competent. A competent means here which is capable of taking the genetic material. So once these cells, the competent cells, are available, then you have to provide the DNA which is from maybe other source. Usually it could be a plasmid DNA which is taken up by the competent cells. They are transferred after transformation. How you will be knowing that exactly these cells are transformed or not? So usually we transform a cell with a, for example, plasmid DNA. It has some selection marker genes, so that when you plate these cells on a selection media, only those cells will be able to survive which has taken up that exogenous genetic material. So you can pick up those cells in a sterile condition and multiply them in an appropriate medium so that you can get a good number of those cells in a culture medium. So basically you have multiplied the genetic material along with the cells. So this is the basic, I would say, the procedure, the simplified procedure for a bacterial transformation process. So based upon this transformation process, the technique of genetic engineering is based on the process by which an organism's genetic material, that is genotype, is modified to get a desired characteristics. So here, what is if the characteristic is visible, then we call visible, or something which is due to that genetic genes which are there in the genotype, genetic material, it gets a phenotype, and this is referred to as genetic engineering. Means you are providing certain types of characters in that host organism which were earlier not available, or if available, you are trying to manipulate its expression level. The new fragment of the DNA which are either inserted or deleted, or what you can also do, you can do editing of target sequence by the process called mutagenesis also. And for the mutagenesis process, you can use different types of chemicals, UV radiations, or transposons. So genetic engineering basically uses a recombinant DNA technology to modify an organism's DNA to achieve a desired trait. So if that trait has been achieved due to recombinant DNA technology or any other similar technology, then it will be called as genetically modified organisms. In short, we also called as GMOs. You can go back and listen my previous lectures on introduction to biotechnology as well as on recombinant DNA technology to get a more clear view on these terms. And the host organism which actually takes up the DNA, a foreign DNA, is considered to be a transgenic organism. It could be a transgenic animal, it could be a transgenic plant, or it could be a transgenic microorganism. So in a simplified way, if we have to explain the procedure of genetic modifications, then the first step should be you have to identify the desired characteristic of an organism. Means how you have to, characteristics is based upon a particular genes. As the central dogma of molecular biology, we know that gene are transcribed to RNA, which is further translated to a protein in certain cases, not necessarily. Then what is the next step? We have to isolate that characters. So how we can isolate the characters means we have to isolate the gene of interest from that organism which is displaying that character or a trait. The next step will be to insert that characteristic into a desired organism. So means here also you are basically putting that gene of interest into that organism, and later on you can propagate that organism. So here you can see that there is no sexual need of sexual compatibility required. Means you can take a gene of interest from totally unrelated organism and you can express it in a totally different organism. So this is the advantage of this technique. Coming to the most important topic of this particular talk is agrobacterium mediated tumefaciation mediated gene transfer. As you know that there are bacteria which are capable of infecting various organisms, but agrobacterium tumefaciation has a special status in genetic engineering, and it is called as natural genetic engineer. Why it is called natural genetic engineer? We will be discussing in this lecture. So, in a natural condition, this particular bacteria is responsible for developing a crown gall tumor in plants by the process which is called horizontal gene transfer. How this actually occurs? This particular bacteria, it transfers its tDNA into the nucleus of the cells of the plant, where it has certain type of injury above the soil, so that this bacteria can sense that there is an injury and it can easily infect that portion. And what it do? It transfers its tDNA into the cells of the plant. Here you can say it's a host plant, and this tDNA gets integrated into the nucleus of those cells. So what will happen? Once this tDNA gets integrated into the nucleus, the genes which are present in the tDNA part also start expressing. And their expression leads to production of certain types of hormones which actually are responsible for the formation of crown gall tumor. So in the diagram also, in this figure, in the slide, you can see here this is a natural plant which is free from any crown gall tumor, but here you can see the development of the crown gall tumor. So this is happening naturally under natural conditions. So what scientists realized, they thought of exploiting this particular phenomena for the benefit of human mankind, and they developed the technique through which we can actually manipulate the tDNA part so that the natural genes can be replaced with the gene of interest which are of importance to us. So before going into the detail, we should know what actually is the Ti plasmid. So if you see this diagram, here you can see there are different types of genes which are written here, like there is auxin related gene for the production of auxin, then cytokinin related, opin related gene. And there is a left border. Here you can see yellow is the right border. And from this left border to right border, this portion is called tDNA region. As explained, this particular region only gets incorporated into the cells or nucleus of the host plant. Outside the tDNA region, you can see there is a virulence region, then there is a origin of replication, and there is a opin catabolism. So there is a function of all these genes. Virulence gene basically is important because it decides or it gives the ability to the bacteria to infect its host. Means if the virulence is more, the more infection can be done. If the virulence is less, less infection is done. So in case of this technology, we rely on a plasmid which is more infectious because we need more transformants. So then there is the region called tDNA region, as explained here. It is this essential region that is transported to the plant cell for infection of the tDNA region, and its length is approximately between 15 to 20 kilobase pair, and it gets integrated into the plant cell via means of genetic manipulation. Then there is opin catabolism. Why this particular gene is important for the bacteria? This region of the bacteria basically helps to provide nutrients for the entire process, and hence it is named as opin catabolism region. So opin can be used as a nutrient via catabolizing sugar phosphates or amino acid derivatives. So there are two different types of opin which are found in TI plasmid. One is called nopalin, another is called octapin type. And last but not least, there is a origin of replication. If you go to my previous lecture, you will find that this particular region is very important for any plasmid to multiply. So if this is not present, then there will be difficulty or replication will not be possible. So origin of replication is also important. So this is the overall diagram figure of a TI plasmid and the role of important genes which are mentioned over here. Now coming to the process of agrobacterium mediated transformation. In this particular figure, you can see that here you have isolated the DNA from your organism. Means this DNA is a particular type of gene, in this case, or gene of interest, which you have isolated, okay, and then you have to clone it. Means in the previous if you see the previous diagram, if you disarm this particular region of auxin, cytokinin and opin and replace it with the gene of interest, maybe selection marker gene, then what will happen? The process will be occurring similarly, but the naturally occurring genes are replaced with the gene of interest. So you have to clone the genes basically. So here you can see that there is a target genes which are cloned into this TI plasmid. So once the cloning is achieved, so your new TI plasmid is formed, where you can see here the red portion basically contains your target gene and the other portion is same as what was there in the naturally occurring bacteria, and so even this portion can also be manipulated based upon the requirement of the researcher. Then this particular complete plasmid, now this is a recombinant plasmid, it is transferred into the agrobacterium cell. So you can see this is a plasmid here, and this is the other genome, normal genome, this is a plasmid, and you multiply the cell, means you multiply this genetic material also. So after the cloning, you multiply them, then plate them on proper selection media, and you will get the cells which are transformed based upon the selection type or selection gene you have cloned into that particular bacteria. Now, what is next? Next, you have to infect your X plant. Now, X plant can be any type of plant tissue which is capable of taking up or can easily be infected by your agrobacterium, which is having your plasmid with gene of interest. So similarly, if your X plant is actually transformed, that also needs to be selected, because how you will be knowing that whether your plant has been has taken up the this DNA or not. Again, you have to plate those X plants on a proper selection media, so that only those X plants will be able to survive on the selection media which has taken up the tDNA region from the agrobacterium. So these are considered, they are grown. Maybe first step is to grow, the shoot generation is there, means shoots will come first, and the shoots which are coming up, we can say that they are putative transgenic plants, because we have not yet checked them through different molecular biology techniques like PCR or southern blotting or any other technique. We are just assuming that the gene has been integrated in these plants. So these are putative transgenic plants. Later on, these shoots are transferred on the rooting media so that you develop the roots, and once these roots and shoots are completely developed, they are hardened in the laboratory conditions, followed by the greenhouse, and they are transferred in the greenhouse and multiplied, and later on these plants can be analyzed for the availability or the presence of the gene of interest which we were looking for, or we were requiring them to be transferred into this host plant. So here is the pictures from my own research work, where you can see this is the callus which is showing some growth of shoots, and later on when the shoot starts growing up, we cut them and we put them on a shooting media so that shoots are developing here. Then you have to cut the shoots, you put it on the rooting media, and then you have to start developing the roots. This is the next step, is you have to harden this plant, because these plants are very delicate, like a small baby. If you just transfer them in a harsh condition, they will not be able to survive. So you need to slowly put them in a harsh conditions, but not too harsh. So we put them in an agro pit. Here you can see the plant is in agro pit, maintaining the proper humidity, light condition and everything. Once you think that they are hardened enough, they are put in the greenhouse condition. And you can see the transgenic plants in this figure. You can see the center one is the control plant without having any gene, whereas the other two plants are the plants which are transgenic plants. So you can see this is a long procedure to be followed to develop a transgenic plant. Now, we have discussed the natural method where we need some organism to transfer this gene. But apart from this, there are certain other methods which are utilized for the transfer of gene. Some of them are like microinjection, electroporation, sonoporation, liposome-mediated gene transfer, and bialistic method. So, coming to the first method, this is called microinjection. You must have seen this type of images earlier also. It is from the image itself you can make out that in this technique basically we introduce the DNA into a animal cell. That animal cell can be a egg, oocyte or embryo, or it can be a plant cell, but in case of plant, we use protoplast. And we use this micropipette, which is the fine tipped glass needles, for incorporating the DNA into the cell. So, what is basically protoplast? Protoplast is the cell which is devoid of cell wall. In plants, we know that there is a cell wall. So, we have to remove that cell wall by a series of procedure, that is enzymatic procedure also, or a mechanical procedure, so that you can remove the cell wall of a plant and it is left with a cell membrane. So, less hurdle, easy transfer of the exogenous genetic material. So, here you can see in this process, which involves incorporation of DNA straight into the either cytoplasm, either in the cytoplasm or the nucleus of the cell. So, this is one of the well-known technique of microinjection, and this is so minute you have to utilize very important instruments, high resolution microscopes, so that you can easily do this procedure without any problem. Next come electroporation. What is electroporation? In electroporation also, you basically permeabilize the cell by the use of electric current. The process of passing through the cell membrane barrier using a brief and high voltage pulses are required in the technique of electroporation. So basically you are using here a type of high voltage but for a brief time pulse, so that the cell membranes develop some types of pores, and hence these pores are big enough so that they can take up the genetic material which is present in the outer surrounding of the cells. So this procedure basically is transient and reversible membrane background. Means once the pores are formed, the material goes inside the cell, and after some times the pore may again go back to the original conditions. So, but the material has been already taken up by the cell. So this is one of the another method of electroporation for direct gene transfer. So this is the instrument which can be used for this particular technique. Next comes is sonoporation. Again, here also in sonoporation, you are using the ultrasound mediated gene delivery system. Means here you are not using the electric current. You are using the ultrasound mediated gene ultrasound. Okay. So this also basically permeabilize means formation of small pores in the cell membrane for incorporation of the nucleic acids. In our case, usually it is our gene of interest. So here also you can see, here is the pore formation, membrane pore formation, and using a sonoporator, this particular gene, means this technique, this picture basically is showing the therapeutic gene transfer. So you can use in animal system or a plant system, but the procedure remains same. And finally, once the pore is formed, your DNA gets integrated into the cytoplasm and it is taken up by the cell. So this is what is called sonoporation. Then another method is called liposome mediated gene transfer. In liposome mediated gene transfer system, you are using a technique of, you can say, endocytosis. Means the cell take up a liposome which is having the genetic material encapsulated within it. So once the cell comes into the contact of liposomes, what will happen? It will go and merge with the cell membrane, and the cell will take up this liposome inside the cell. And once the liposome is inside the cell, what happens? It will break down and the genetic material will be released inside the cell. So you can see here we are using this important technique of liposome mediated gene transfer for direct transfer of the genes. Here comes another important technique which is widely used in plant biotechnology, that is called bialistic method. What is bialistic method? From the name you can make out it is having something to do with biological ballistics, means it is a type of gun, but this gun is not a regular gun, it is considered called as a gene gun. So this word bialistics basically is a short form of the original word which is called biological ballistics, which is also known as particle mediated gene transfer system or also called as gene gun. So here also in this method we directly shoot the DNA coated small particles. These small particles can be tungsten particles or it can be a gold particles also, which are coated with the DNA which is having our gene of interest, and they are bombarded on the cells or the tissues which needs to be transformed. So this is also one of the widely used technique for transformation of plants and other living forms. Here you can see this is the instrument which is called the gene gun, or which is used for transformation of these plant cells or animal cells. So in short, if we have to define this procedure, this technique basically involves coating high density gold or tungsten particles, here they are called as microcarriers, with nucleic acids or other biological molecules. But the question comes that in a normal gun you usually see there is some type of inflammable material which gives that energy that the bullet goes and hit the target. But what is here? How this energy comes from where? In this particular gene gun, the helium pulse is basically used to accelerate the microcarriers to a high speed, which allows them to pass through the cell walls or the cell membrane also of the target tissue or a cell. So in this case you may use a complete plant cell also, which not necessarily a protoplast. So this is the advantage of using this particular type of direct gene transfer system, because in the previous one there was a like in some techniques you have to use a protoplast. Here in this diagram you can see that here you can see the gas is accelerated through the tube. This is the tube. Okay, here you can see this is the gas which is accelerating through the tube. Then there is a rupture disc. Rupture disc is put there because how we will know whether there is a hit by a helium gas or not. So if there is a proper pressure, certain pressure has to be maintained. If that proper pressure is maintained, what will happen? This rupture disc basically breaks. And then you can see here, you can see there are DNA coated particles. So what will happen? Once the gas is with a particular pressure is supplied, this rupture disc basically gets broken, and this DNA coated particles, they hit a base. Here you can see a base which is called stopping screen, means it will stop the base which is holding your particles. So it will hit this stopping screen, but this stopping screen is not that big enough to stop those particles which is having that DNA or a gene of interest. And in the last you can see here is your tissues or the cells which are lying, and these coated particles, they hit these cells or the tissues, and finally some of or a good transformation efficiency is found, these particular cells and tissues take up that exogenous genetic material. And then again, as I have described for agrobacterium also, here also you have to go through the same selection procedure, and then you select the putative transgenics plant for further analysis. And now there is lot of advancement has been done in this technique. You can see here a representative picture for a commercial portable gene guns also. So you can see that the principle of the technique usually remains same, but with the advancement of the technology, things become more easier and more advanced. So same applies to the biologistic method. So we'll take a short break here, and after the break I'm going to discuss an important technique of CRISPR-Cas9, which is also a technique for genome editing. Thank you.
A warm welcome to IGNOU. If you're creative, if you can write well, and if you can visualize, mass communication is the correct career option for you. I am going to talk about postgraduate diploma in journalism and mass communication, which is also the first year of MA program in journalism and mass communication. So any learner who has done a postgraduate diploma in journalism and mass communication is eligible to take admission straight away into the second year of our master's program in journalism and mass communication. Duration to complete this program is minimum 1 year, and because we have this inherent flexibility in distance education system, you can take up to 3 years to complete this program. The minimum eligibility criteria for taking admission in this program is bachelor's in any discipline. To give you a glimpse of what are the various components of this program, we begin with telling you what mass communication is, what are the various areas, various branches of mass communication. Then we go on to tell you about the various reporting techniques and beats of reporting, followed by media ethics, the principles, the codes of conduct that you must follow, and we also focus on the writing skills and editing skills for print media. Now this program comprises of 8 courses of 4 credits each. 25% of the entire program is practical based. So we have 6 theory courses and 2 practical courses. This program is so designed so that it will be able to make you more employable in the market. So it helps you build a career in print media, in radio, television, new media, as well as the allied subjects of mass communication, which could be media marketing, advertising, public relations and corporate communications, event management. This can also help you become an entrepreneur in areas like photography or having your own production house and so on. So I hope that you will be excited to join this program, and we will be waiting to welcome you here in IGNOU.
Welcome back to Gyan Darshan. Before this short break, we have discussed about the method what is called transformation, followed by bacterial transformation, agrobacterium mediated transformation, and some of the methods for direct gene transfer of any gene of interest. Now, we are going to discuss a very important technique of CRISPR-Cas9. You must be knowing that we as a human have an immune system to cope up with the infections caused by various microorganisms, maybe virus, bacteria. But have you known, do you know that how basically these microbes, they protect themselves? Recently, whole world has suffered due to COVID. The causative organism was the virus. And these viruses just not infect humans. They are capable of infecting bacteria too. So, to understand the CRISPR-Cas9, we have to understand basically how bacteria protects itself from the infection from a virus. So let's get back to CRISPR-Cas9. So in starting, it was thought that CRISPR system is actually a repair mechanism of a bacteria or an archaea. But later, the scientist Mojica and co-workers in 2000, they noticed that there are certain spacer sequences which are similar to the sequences which are found in bacteriophages. Bacteriophage means the viruses which are capable of infecting the bacteria. And these particular sequences, spacer sequences, were also found in viruses and plasmids. So this was a hint that why it is now found, these particular sequences are found in all these organisms. So they discovered that viruses cannot infect bacteria who possess these homologous spacer sequences. Means those bacteria which were having these sequences have a protection from infection from the virus. And this gives or suggested that there are certain role to play by these sequences in adaptive immune system of a prokaryotes. In 2007, Barong, along with his co-workers, they published a paper where they showed that there is certain new spacers which are derived from the genomic sequences of the phages were integrated into the virus-challenged bacteria. Means there were certain sequences which were already present in the viruses were also found in the bacteria which were previously infected by those viruses. So they also demonstrated that by removing or adding these particular spacers, they can actually alter the resistance of that particular cell. Here in this case it is a bacterial cells against the virus. So this was a short background that how this CRISPR-Cas9 story started. So the CRISPR-Cas9 is basically, you can say, is a genomic editing tool which is based upon bacterial adaptive immune response system of prokaryotes, or in particular you can say here in this case was bacteriophages. So there were certain CRISPR sequences which were derived from the viruses who have infected the prokaryotes, and it was observed that bacteria captures those leftover DNA from the invading viruses. Means initially any virus has infected that bacteria, and as we know that viruses hijack the machinery of the host usually, so they leave their genetic material, but bacteria being more smarter in this particular case, it captured that leftover DNA from invading viruses. And what it did, it created the segments of CRISPR arrays. Means it utilized those information which is hidden in the genetic material of that particular virus which was left over, and it prepared a CRISPR array. And when the virus next time, if it infects the bacteria, what the bacteria do? What it started do? It started transcribing a short CRISPR RNA, in short which is called as cRNA, that can guide the CRISPR associated Cas protein to cleave complementary DNA or RNA of the virus sequences. So here you can see that bacteria itself has learned from the virus that how it can utilize its own information, the virus information, to neutralize the virus during the next, you can say, attack on the bacteria. And depending on the type of CRISPR-Cas system, the virus infection can be eliminated in the host or bacteria. So CRISPR sequences, you can say, are used as a guide to recognize and cleave specific strands of DNA which are complementary to the CRISPR sequences. So this information was basically, you can say, exploited or utilized for the human beings for developing this system. So here in the picture you can see, first you have to understand what is CRISPR. So CRISPR is basically described as a region of DNA which is made up of short repeated sequences with the so-called spacers sandwiched between each repeat. So you can say there are different repeated sequences. So each repeated sequence, after each repeated sequence there is a spacer, then again there is a repeat similar sequence, then again spacer, and this is called a type of array. Then comes Cas9. This Cas9, you can say, is a CRISPR associated sequence which is a dual RNA guide DNA endonuclease enzyme that acts as a scissor to cut a DNA. So in my previous lecture also I have explained how endonucleases basically act as a molecular scissor. So this is a type of endonuclease which is also utilized for cutting a DNA, but this is regulated by CRISPR. So that's why it is considered in combination, that is CRISPR-Cas9. But any system has to be controlled also. For that there is a role to play by another important molecule which is called PAMs. The full form is protospacer adjacent motifs, which are short 2 to 6 base pair sequences in a viral genome which are found adjacent to the sequences targeted by the Cas nucleases. So in this diagram, if you see that if this is the sequence which has to be targeted, then PAM is helping that the neighboring sequences should not be targeted. Only the sequence which has to be edited or replaced should be cut by the or cleaved by the Cas9, but not the other sequences. So it has a protective role in this particular case to play. So PAMs play an important role in ensuring that only foreign viral nucleic acid are cleaved, but not the CRISPR arrays. So this PAM plays a very important role, that this CRISPR-Cas9 system should work without any type of non-specific editing. So when this complete system, considering the CRISPR, Cas9 and PAM, is introduced into a host complex organisms, it acts so nicely that it can be utilized as a genetic editing tool. So here you can see in the diagram, this is the target sequence, and based upon the target sequence, the CRISPR array is designed, and based upon the sequence, the Cas9 is also utilized and PAM is there. So finally what it happens, it removes the targeted sequence, and finally what happens, this pre-designed DNA is integrated here in this particular site, and the previous section is removed. So you can see that compared to the previous system, this system do not use that long process of selection, but this is more targeted, and in case of agrobacterium mediated transformation, it is random process where this process is more targeted, and hence this system can be considered as more advanced compared to the previous system. And this particular technology basically has been used also in many researchers these days. So if you have to see the timeline, it's not very old. In 1987, first discovery of CRISPR was done in E. coli. In 1993, discovery of CRISPRs in Mycobacterium tuberculosis was done. In early 2000s, the adaptive immunity role of CRISPRs were discovered. Then in 2012, CRISPR-Cas9 for the use of genetic editing tool was used, and the publication in the journal Science names CRISPR's breakthrough of the year was published in 2015. In 2020, very recently, the Nobel Prize in Chemistry was given to Emmanuel Carpentier and Jennifer Doudna for their development in this powerful gene editing tool. And beyond, this particular technique is right now used for human mankind in curing many of the diseases. Let's just quickly have a look on the application of CRISPR-Cas9. This CRISPR-Cas9 system can be used as a cut and paste tool, as I have explained, to modify the genome using the guide RNA. Means you design the RNA in such a way that it goes and replaces the previous, the target region only, and with a new gene, a gene of interest. Then this particular technique can be used even for increasing the lipid production in microorganisms like microalgae, for increasing the yield of biofuels also. This can be used also to remove or to introduce new genes, as well as to silence or to activate these genes. So we can use different types of manipulation. You can use dead Cas9. Dead Cas9, D-Cas9 is a dead Cas9, so it will not be doing its job. So in combination with the CRISPR, you can use in such a way that it can go and do the targeted mutagenesis only. It will not replace, it will mutate the gene at a particular location. So you can either activate a particular gene by using CRISPR-A, or to inhibit a particular type of gene by using CRISPR-I, using this D-Cas9. Then you can also use this particular, as I have explained, for targeted epigenome editing. You can use this particular editing tool to mutate different types of enzymes like DNA methylases, acyl transferases, by cutting at a particular position. And this technique has been widely used for recently also for the cure of genetic disorders like sickle cell anemia, cystic fibrosis, or disease like cancer or infectious diseases. In 2019 itself, this was used for the patients suffering from sickle cell disease and beta thalassemia. So this is a promising tool for many of the people who are suffering from these diseases, and as this particular technology is a latest one. Many new changes will be coming up, and we hope this technology can be a turning point for a human mankind. So, in today's lecture, I have discussed different tools which were traditionally used in genetic engineering, ranging from agrobacterium-mediated transformation to direct gene transfer to the latest technique of CRISPR-Cas9, which is widely used nowadays for genome editing. So, with this, I'm going to end my lecture for today, and I hope if you have any type of questions, you can mail me on my email ID, which was displayed on the first slide. I will be happy to respond to you all. Thank you so much.